Hi everybody, and welcome to the latest VodCast for Honors Biology at Desert Ridge High School. I'm Mr. Galladay, and today we're going to be talking about natural selection as an idea since the time of Charles Darwin. This is a good place to update your notebook organization because here we go. First, it's a good idea to review just what we mean by a theory. Well, you should recall that a theory is an explanation that pulls together facts, laws, observations, and other tested hypotheses. When we get new legitimate evidence that contradicts a theory, that theory is either modified or discredited, as was the case with Lamarck's theory of evolution by acquired characteristics. That theory was no longer accurate once we understood how traits are passed from parent to offspring. Well, Darwin published On the Origin of Species in 1859, which was over 150 years ago. And today we're going to see if Darwin's theory is still supported by new discoveries or if it should be discarded along with Lamarck's theory. Well, since Darwin's time, there have been new discoveries in four main areas that relate directly to natural selection: genetics, new fossil discoveries, molecular biology, and a new science of genomics. Genetics, molecular biology, and genomics are all branches of science that did not exist in 1859. So, if natural selection is a flawed theory, you would expect that these new branches of science would expose any flaws. And that's what we're going to see today. Okay, so first we'll look at The Origin of Species, as we said, was published in 1859, and Mendel's results were published in 1865. Now, we know that Darwin was not familiar with Mendelian genetics because he confessed to not knowing the mechanism for how traits are passed from parent to offspring. Now, modern genetic theory explains how populations maintain genetic diversity, which, as you know, is needed for natural selection to act on. Remember, natural selection requires a population to have genetic variation. Ideas such as recessive genes, meiosis, crossing over, mutations, and multiple alleles all contribute to genetic variation in a population. So modern genetics supports evolution by natural selection. Now we're going to take a quick look at the example of Chaetodipus intermedius, or the rock pocket mouse, that lives in rocky habitats of the southwestern U.S. and northern Mexico. Populations of these little guys tend to be small and isolated, and previous experiments show that the dark phenotype is caused by mutations to a particular gene called MC1R. Well, this gives us an opportunity to directly test Darwin's theory using a science that did not even exist in 1859. We want to find out if relative allele frequencies of the MC1R gene change in populations living on different colored rock habitats, and then if so, are these changes due to natural selection, or is it because individuals are migrating? In other words, is this little mouse saying, holy cow, I got to get my mousy butt off of these white rocks and over to a black rock, or I am going to be coyote food? And the white mouse is sort of thinking the same thing, that he's got to, are they migrating, or is it because the populations are evolving? Well, the first question was answered in a study done in 2003, showing a strong selective pressure and correlation between mouse fur color and the color of rocks where they lived. You can see here that all the light-colored mice live on light-colored rocks, and the dark-colored mice all live on these dark-colored volcanic rocks. Later tests showed that the dark populations arose by unique mutations to the MC1R gene, not because individuals were migrating. So what this means is that these dark-colored guys were arising in populations independently of each other. They weren't moving to darker-colored rocks to match their fur. So these experiments confirm that different selective pressures can cause changes to a population by the process of natural selection. Well, now we're going to take a look at a new fossil discovery to see if the fossil record in the 21st century leads to the same conclusions that it did in 1859. Well, the first thing to understand about the fossil record is it will always be incomplete. And this is because the conditions needed to make fossils are pretty rare. Conditions to produce fossils are much more common in aquatic environments than they are in, say, desert environments. Here we see a dead crocodile at the bottom of a river. Okay, his body gets covered up with sediments as the soft tissues decay, and this leaves the harder tissues such as bone behind, which gradually get chemically changed into rock over millions and millions of years. Now, if the upper layer gets eroded to expose our crocodile, then somebody can come along and find a really cool fossil. If the upper layers don't erode, then we have a fossil that is buried under miles of rock and sediment and can't be studied. So all of these situations you can see are rather rare. So the fossils that we have, I guess you could say we're quite lucky to have them. Well, we have two techniques to determine the age of rocks and the fossils that are sometimes in them. This is a course in biology, not geology, so I'm not going to spend a lot of time explaining geological terms. You should, however, know the difference between absolute and relative dating. Okay, the first technique is called absolute dating because we use an absolute molecular clock, and that clock is the known time for some isotopes to decay. And that is to say, to determine how long it's been since a particular rock was molten. This technique is useful for igneous rocks, but it's not useful for sedimentary rocks, and unfortunately sedimentary rocks are where we find the fossils. The second technique is called relative dating, and this is not going to the movies with your cousin. This is a technique that we use to determine the age of sedimentary rocks. Now we can compare layers with the same fossils to other layers where a time can be established with absolute dating techniques. So here we've got a lava flow which is igneous and granite which is igneous, so we can get an absolute date on the layers above and below this fossil. Now since we have the same fossils here that we do over here, we can infer that these are the same age, that these two layers are the same age. That's what we mean by relative dating. Well, using these techniques with hundreds of thousands of fossils pieced together from all over the Earth has allowed us to create a geologic timescale. This timescale has been pieced together like a puzzle by thousands of scientists over the last 200 years. This is a greatly simplified version, but it does show one area that has tantalized paleontologists for a long time. The early Devonian period has lots of bony fish, including fish that had thick bony fins called lobe-finned fish. Now by the end of the Devonian, we also had four-limbed animals that were walking on land. Okay, here's a little more detail. Well, first we can see that there's lots of lobe-finned fish in the early Devonian. Suddenly, well, if you can call 30 million years sudden, the fossil record shows four large tetrapods, or four-legged tetrapods, walking around on land. If Darwin's correct, there must be some transitional species between the fish and the tetrapods. Okay, so this gives us a testable question. Well, if we look at the exposed rock of the right age, which is about 375 million years old, that came from the right location on Earth, namely a place that had a tropical climate 375 million years ago, and was in an estuary or tidal flat, that is to say a river system that has shallow muddy water, well, we should find a transitional species, something that's not really a fish but not really a land animal either. Okay, something Darwin didn't know is that the continents were not in the same locations 375 million years ago as they are today. This is because plate tectonics was not established until the middle of the 20th century. Well, in 2003, scientists found such a place. They found a field site in extreme northern Canada that had very different environment in the Devonian period. Well, here you see the place in northern Canada. Here you see where it was during the Devonian, 375 million years ago. Well, it had exposed rock that was the right age. The climate in the Devonian had been warm and tropical, and it had been in a system where rivers dumped into a tidal flat at the time. So if Darwin's ideas and the ideas of modern geologists are correct, we would expect that there might be fossils of a transitional species at this location. Well, in 2004, scientists discovered fossils with the following observable characteristics. First of all, these things had traits of fish such as scales, fins, very weak gills. It also showed signs of having lungs, such as many other lobe-finned fish at the time did. It also had some four-legged tetrapod traits, such as a distinct neck. It had ribs. It had a very flattened alligator-like head with eyes on the top, not on the sides as they are with fish. and the limb bones had structures that were recognizable and common to all of today's tetrapod species. So this is to say a humerus bone, a radius, and an ulna, wrist bones, and even digits. The fossils also had ear notches in the skull, which were more like a land animal and less like a fish. So here's a reconstruction of the animal based on several intact skeletons. The first 3 skeletons were discovered in 2004, and they named this species Tiktaalik rosier. Well, since 2004, over 30 more individuals have been discovered in the same area of northern Canada. The discovery of Tiktaalik in the exact location predicted by our current knowledge of geology confirms both Darwin's theory and the geologic theories that allowed us to locate the fossils in both time and space. Okay, our next area of new discoveries is the area of molecular biology. Now, this deals with molecular structures within cells, such as DNA, genes, proteins, and other complex molecules. It's pretty safe to say that this entire branch of science wasn't really in the same form that it is now that it was as back in 1859. Our testable question comes about from a couple ideas. First of all, our current model, based largely on fossil evidence, suggests that modern birds are direct descendants of some extinct dinosaur species, right? So here we have our modern birds, and from fossil evidence, it suggests that they are evolved from the ancestors of some dinosaurs. We also know that living organisms often have sections of DNA that aren't used. Some of these unused sections of DNA appear to be deactivated genes. And in addition, we know that modern bird species don't really have true teeth, such as many of their dinosaur ancestors did. So here's our question. If modern birds evolved from the same ancestors as dinosaurs, then there may be unused genes in modern birds that were expressed by dinosaurs. Well, were there? Well, in 2006, researchers activated one of these unused genes in a chicken embryo. They turned on and activated an inactive gene, and the result was an embryonic chicken that began growing very reptilian teeth. Okay, on the left you can see our normal chicken embryo contrasted with the chicken with the activated gene on the right. So again, we have evidence from a science that did not exist in Darwin's time, supporting, not refuting, the idea that species have changed over time. Okay, our fourth and final area of science during Darwin's day is a new science which we call genomics. Genomics deals with the entire genome of a species, that is to say, the entire genetic library. All of the 3 billion A's, T's, C's, and G's that make up you are your genome. Because the enormous amount of information, genomics is a blending of biology and information technologies that obviously did not exist in 1859. Well, you may know that the human genome was sequenced in 2001. You may not know that the genome of the chimpanzee was completed in 2005. Well, we have strong fossil evidence and also anatomical evidence that suggests our closest living species is chimpanzees. That is to say, we both evolved from a common ancestor who lived about 7 million years ago. So this is our timeline going back about 7 million years, and this is when humans and chimpanzees and bonobos all split off. Now, I want to be clear here, humans did not evolve from monkeys, apes, or any other presently living species. They are our evolutionary cousins, not our grandparents. So one aspect of this common ancestry that's puzzled biologists is the observable fact that we have 23 pairs of chromosomes, but all living ape species have 24 pairs. Now, it's not possible that humans lost a chromosome. The loss of that much genetic information would be fatal. The most likely explanation is that one of the human chromosomes was formed by a fusing of 2 earlier chromosomes. Well, this forms a testable question. If humans and chimps share a common ancestor, then our genome must show signs of a fused chromosome. Well, does it? Well, first thing we want to know is, what would a fused chromosome look like? Well, to see that, we need to know that every chromosome has a recognizable section called at each end called a telomere. Chromosomes also have recognizable sections in the middle called a centromere. you may remember that centromeres are the location where sister chromatids attach before anaphase in mitosis. Anyway, this leads directly to our testable question. If humans and apes have a common ancestor, there must be signs of a fused chromosome in the human genome. That would be a chromosome that looks kind of like this, that has a telomere at either end, as well as 2 telomeres in the middle and 2 centromeres. Well, the mystery was solved with the new science of genomics by comparing the human genome to the chimpanzee genome. Human chromosome 2 matches up exactly with chimpanzee chromosomes 12 and 13. In 2005, scientists compared human and chimp genomes and located the precise spot on human chromosome 2 where the fusion took place. Again, we see an example of new discoveries confirming Darwin's ideas that populations have changed over time. So let's quickly summarize all of this. Well, when new evidence comes along that challenges an existing theory, that theory is either thrown out or modified if the new evidence is shown to be repeatable and believable. Mendelian genetics and our current model of inheritance were found to strongly support natural selection, but not Lamarckian evolution. Lamarck's theory was discarded, but Darwinian theory persists because it is supported by the evidence. New fossil discoveries have been able to pinpoint expected locations of transitional fossils, not only with Tiktaalik, but also with transitional species between modern whales and their land-dwelling ancestors. Molecular biology continues to make discoveries that use natural selection as a powerful model that makes correct predictions. And then the new science of genomics has confirmed Darwin's concept of the tree of life by showing that all living things contain the same genes, and these genes have evolved new uses over time. In short, new scientific discoveries have been unanimous in confirming and expanding on Darwin's ideas, first published in 1859. It's the mark of a strong theory when it can withstand harsh criticisms and new unforeseen technologies but remain useful. Well, that's it for this VodCast. I'm Mr. Galladay for Honors Biology at Desert Ridge High School, and I hope you have a great day.